What is light reaction? The light reaction is a part of photosynthesis in which objective is to generate a assimilatory power so that they can be utilized for the fixation of atmospheric CO2 in the dark reaction. This is Dr. Pankaj Kumar, your mentor, and in this session we will be talking about light reactions. So light reactions, we know very well that occurs in case of grana. The raw materials required are light as well as water. Water is oxidized into oxygens and the assimilatory power is going to be formed in the form of ATP and NADPH2. So we will just learn that how it is going to be formed, right? So first of all, let's talk about the mechanism of the light reaction, and we can divide into two parts. One, the photochemical phase, and second, the photophysical phase, or we can say it's a light reaction. Even the photochemical phase includes just one thing, that is photolysis of water, and in case of light reaction we will be talking about non-cyclic and the cyclic one. So first let's talk about the photolysis of water. Now photolysis of water, what does it mean? That the water under light is going to be broken and finally oxygen is released. Earlier it was a big controversy that what was the source of oxygen? Is it a water or is it a CO2? But it was a Rubin experiments which eventually proved that the source of oxygen is the water, not the carbon dioxide. Okay. So the photolysis is going to occur in the intrathylakoidal space, right? And it is associated with the PS2, and the three elements is required, what we call manganese, chlorine and calcium. So what happens? First of all, the water molecules is broken, then what we see that 4 hydrogen ion and 4 hydroxyl ion is formed. Now this 4 hydroxyl ion breaks into 4 electrons, and finally this produces a water molecule as well as oxygen is actually being released. So this is how the photolysis of water is happening, right? Let's talk about Hill's reaction or the non-cyclic pathway or the light reactions. Now what is a non-cyclic pathway? In case of non-cyclic pathway, what happens? First of all, that both PS1 and PS2 is involved, right? Here photolysis occur, so that's why oxygen is released. And why we are calling them non-cyclic? Because the electron lost by PS2 is compensated by the electrons coming through the photolysis of water, and similarly electron lost by PS1 is compensated not by its own electron but the electrons coming from PS2, right? So that's why we are calling them the non-cyclic. Okay, let's see that what are the sequence of events during the non-cyclic electron transport. Right now you can see there are two photosystems, P700 and P600. So under photo excitations, what happens? They will lose an electron, right? But in case of photosystem 2, the electrons being compensated by water, as you can see there, as a result of photolysis, the electrons will be formed and electrons will be finally going to the PS2, and thereby their loss of electrons being compensated over here. But the electrons which is expelled by PS2, it is accepted by phosphoitene. Now phosphoitene is a very important. Why? Because this is the only photosynthetic intermediate where magnesium is not there, right? In rest of all photosynthetic intermediate, magnesium is there. Okay, from there it goes to plasterquinone, right? And from plasterquinone it goes to cytochrome. You see, cytochrome, there are different types of cytochrome, right? Cytochrome F, cytochrome B5, F6. So depending upon the type of a species, there are different types of cytochromes are there. Okay, from cytochrome it goes to plastocyanine. Now what is plastocyanine? In case of plastocyanine, what happened? The central material is copper. Many a time this question has been asked in NEET. Okay, so here what we see, there is a central material is a copper, but in case of cytochrome there is a iron. Even in case of plasterquinone there is a iron, but in case of phosphoitene, what we have seen, magnesium is absent, right? From plastocyanine it goes to P700, and there we see that the loss of electron by PS1 is actually compensated by the plastocyanine. From PS1 the electrons are accepted by ferrodoxin reducing substances, right? And from there it goes to ferrodoxin, okay? And then this electron being accepted by NADP ion, okay? Now what happens? This is joined by the hydrogen, okay, and as a result NADPH is going to be formed. This is one of the assimilatory power, right? So this is how the assimilatory powers are formed in case of the light reaction or the non-cyclic electron transport system. So what we say, that in case of non-cyclic electron transport system, that assimilatory powers are formed in the NADP and ATP. How ATP is formed, we will discuss in chemoosmotic hypothesis later on, right? Let's talk about the cyclic one. But before cyclic, let's discuss one more interesting thing, what we call Plasterquinone pump. Now what is Plasterquinone pump? The very function of the Plasterquinone pump is that they pick up hydrogen ion from the stroma and pump it to the intrathylakoidal space, and thereby they generate a electrochemical differences between the intrathylakoidal space and a stroma, and that is the reason that the ATP is generated. Okay, let's see what are the sequence of events during the Plasterquinone pump. So this is a stroma, this is a thylakoidal membrane, this is an intrathylakoidal space, right? And this is a ATP synthetase. Now we know very well that photolysis occurs in the intrathylakoidal space. As a result, electrons are released. Now these electrons are going, right? You can see there that Plasterquinone is accepting these electrons. So the moment it accepts electrons, it becomes PQ minus. So here we can say that this is the PQ minus, right? It goes to the up, it picks an electron, so now it becomes PQH, because we know very well H ion plus electron, this will become PQH. Now it comes inside, okay, now this will release H ion inside. So here what happens, it releases H ion and again it gets converted into the PQ minus. So again this is a PQ minus, okay, and thus again it goes back to the PS1. So you see that this molecule keep on moving from the outer end to the inner end, and thereby picking this H ion and transferring into the intrathylakoidal space, and this particular event is actually called as a Plasterquinone pump. Okay, mind it, there are two sources of H ion in the intrathylakoidal space. One is the photolysis of water and second is the Plasterquinone pump. Now what is the fate of this H ion? You know here that slowly the concentration of H ion will increase. Okay, this entire inner membrane will not allow the passage of H ion. So definitely here what will happen? pH will become very, very less or acidic. Okay, there is only one place from where the hydrogen ion can cross, right? And that place is what we call ATP synthetase. So just see, the hydrogen ion is crossing. So the moment hydrogen ion cross, energy is released, and that energy is utilized for coupling of ADP inorganic phosphate, and as a result ATP is formed. Okay, later on this H ion moves on and it binds with NADP and the electrons are coming there, right? And finally assimilatory power in the form of NADPH plus H plus is formed. So just see here what we see, both the ATP and the assimilatory power in the form of NADPH or NADPH2 is formed, right? So this is all about the formation of assimilatory power and the mechanism what we have. Let's talk about cyclic electron transport pathway. So in case of cyclic transport pathway, what we see, only PS1 is involved. So you can see there that here the electrons being expelled, it is accepted by ferredoxin reducing substances. Okay, so instead of going outside, again it is accepted by Plasterquinone. So here what happens, Plasterquinone pump is operating, right? And since Plasterquinone pump is operating, that's why it is responsible for the formation of ATP, right? So what we see in case of cyclic electron transport, only PS1 is involved. There is no photolysis. Loss of electron by PS1 is compensated by their own electron. That's why we are calling them cyclic. PQ pump operates and assimilatory product is formed only in the form of ATP, right? This is about cyclic electron transport pathway. Now let's talk about the energetics. Now in case of light reaction, what we see that one quanta or photon of light is required to move one electron. So naturally, for the release of one molecule of oxygen, 8 photons or quanta is required, 4 by PS1, 4 by PS2. Okay, and 18 ATP and 12 NADPS2 are formed as a release of 6 molecules of oxygen, and this relation is very, very important in solving some of the problems, right? We will see that how we can use this relation for the formation for solving some of the problems. So once again, I would like to reiterate that 8 quanta of lights are required for fixation of 1 molecule of oxygen. Similarly, 18 ATP and 12 NADPH are required and as a result 6 molecules of oxygen are released. Let's try to see some of the questions which is based on these relations. Now the question is, what is the number of ATP formed during the release of 30 molecules of oxygen in light reaction of photosynthesis? Right now we have seen that altogether 18 ATP is required for the release of 6 molecules of oxygen, right? So obvious, for 1 molecule of oxygen, what is the requirement? Requirement is 18 by 6, that is 3 ATP. Okay, so for 30 molecule of oxygen, the requirement is 3 into 30, that is equal to 90 ATP, right? So the correct answer of this particular question is 90 ATP. Okay, let's see one more question, that what is the number of photons required by PS2 for the formation of 4 molecules of oxygen? Again, we will utilize the same relation, because you see 8 quanta of light is required for the release of 1 molecule of oxygen, right? So naturally, for 4 we need 32 quanta, but mind it, this 32 quanta was divided like 16 for PS1 and 16 for PS2. So the correct answer of this particular question is 16, because it is asking only for PS2 only, right? One last question. What happens in bacteria? The oxygen doesn't release during the photosynthesis. What is the reason? Very simple. They don't have a PS2, because we know very well that release of oxygen is always required the presence of the photosystem too, right? That's all what I have to discuss as far as the session of light reaction is concerned. Thank you.